Host defenses. Build lecture number 4, chapter 16. Destroying antigens. Phagocytes. Phagocytes are types of white blood cells whose job it is to protect the host from pathogens that would otherwise cause the host harm. They're what I like to call warrior cells. They literally will hunt down, capture, and destroy. Types of phagocytes. There are 3 main types. We have neutrophils, and these react early when inflammation first arises, and they create pus, which is like, everybody knows what pus is. It's like yellow and white, and it comes out of, big lumps when you squeeze. Next are monocytes. These are activated by chemical stimulus. They transform like magic and become macrophages. Macrophages are basically like, if you ever watch Dragon Ball Z, Super Saiyan versions of monocytes. Next, we have the process of actual phagocytosis, which I have demonstrated over here on this picture to the right. Phagocytosis is the process of swallowing antigens, phagocytes, and then digesting them. In other words, they seek out and destroy invading antigens that wish to cause harm to the human host. One way the human immune system protects the body from infection is through phagocytosis. Phagocytes and what they do. The term phagocyte means eating cell. They will literally hunt down antigens, launch an attack, engulf them, and then eat them. They use a multitude of antimicrobial substances in order to kill them. The steps of phagocytosis are as follows. 1 is chemotaxis. This is where they actually draw the antigen or pathogen, however you want to say it, into their self. Then they will ingest. That's step 2, ingestion. They'll form a vacuole that will surround the antigen, kind of like, an envelope thing. It'll surround it just like a big blanket would surround you in the winter when you're cold. This is the swallowing process happens here. Then you have the phagolysosome formation. This is when lysosomes become one with a phagosome, and this results. The 4th step is destruction. This is the combo of enzymes and toxins released to kill and destroy the pathogen. And then the last step is excretion. During excretion is when what's remaining of it, the dead pathogen, is pushed out, and then it will be absorbed by the body and then exit the host, but it will no longer be alive. Next, we have inflammation. What causes inflammation? Inflammation is caused by post-trauma from an infection. It can also be caused by injury to tissues of the body, such as if you're doing dishes or something per se and you wash them by hand, a knife accidentally cuts your hand when you pick it up. That's going to cause inflammation set up, and you're going to have probably redness happen at the site of the cut, and this is when, the body's defenses are going to start coming together to send platelets and stuff to that part of the body to where they will heal that cut. What else causes inflammation? Necrosis, which is just death of the tissue. This can be caused by physical injuries or ones caused by chemicals. Another thing that causes inflammation is adaptive immunity situations, which I'll be going over those in the next Build a Lecture. What is the purpose of inflammation? The purpose of inflammation is to attract and bring together immune response cells to the site of the injury. This is, as I said before, platelets. Anytime there's an injury, platelets are going to be notified. They're little red blood cells that will all come together and just start piling up on each other in order to close the wound. The process of inflammation, step 2, is to begin the process of repairing the actual damaged tissues. This is again where the platelets begin doing their thing. They were going to find and remove any harmful substances from the wound to keep it from getting infected. And then they will obliterate or destroy microbes and roadblock them from returning to cause harm to the human host. Fever. Fever is caused by inflammation or by infections. Fever is just an abnormally high base body temperature due to injuries, illnesses, cancer, allergies, infections, and many other types of illnesses that are not listed. Temperature is regulated by the hypothalamus, which is a section of the brain. It's literally the body's thermostat. It sets the base temperature for homeostasis. Homeostasis, again, is the equilibrium of all the functions of the body combined that keep us alive. It cools the body during high temperatures. It sends signals to warm the body during cold temperatures. The way that the hypothalamus warms the body up is through shivering. When you shiver and you're cold, that's your body's process of trying to bring up your base temperature back to that 98.6. When you sweat, that's when you're too hot. That's your body's way of cooling back down to a normal temperature so you don't get too hot, which that can cause a slew of other problems, which we don't really have time to discuss right now, but if they're not good. The regular body temperature, I've always been told, is 98.6, but my own body temperature is only 96.6. Typically, when I take my temperature, that's my base temperature. When I have a temperature of 98.6, I'm actually technically running a small, slight fever. But it is normally thought to be 98.6 by most people. There are benefits of fever. Fevers prevent temperature-sensitive pathogens from multiplying and causing more problems. So if there's a high fever, that process is going to stop right now. And if they can't multiply, they can't make you sick. Benefits of fever happens, in instances of diseases and illnesses such as poliovirus, cold viruses, certain fungal pathogen viruses, and infections. They help a person to get better faster in some cases. If you wait until the fever breaks, most time with certain illnesses, you'll feel better. It cuts down on the amount of iron that is released into the body because bacterial pathogens need iron. It's their food source. They cannot multiply without a food source. So the warrior cells that are sent to, help with the whole fever process, they actually cut down on the iron so that the bacterial pathogens are not fed. Fever stimulates the body's immune response. It causes phagocytosis to initiate, which is with phagocytes. They increase metabolism of the body, and it also increases adaptive immune reactions, which again I will be discussing more thoroughly in the next Build a Lecture 5. Should fevers be stopped? This is always a good question. When we get a fever, should we take medications to lower it, or should we just let it run its course? That answer is not always black and white. Sometimes it's hard to tell, but sometimes yes, sometimes no. It really just depends on what the illness is. In the following cases I'm about to describe right here, the answer would be no. And that's when the fever is super high, like really high. I think it's usually anything over like 103. When you start heading to 104 degrees, you need to get that temperature down immediately. Also, if the temperature is, like if your fever is lasting for a super long time period with no reprieve, like tepid baths are not bringing the temperature down, just anything natural like cooling cloths and stuff are not bringing it down. If you happen to take something like ibuprofen or something and it doesn't stop it, then you need to go be seen by a doctor. If a person has any of the following conditions, it should also not be allowed to continue. If they have heart conditions, any kind of head injuries, history of seizures, any respiratory conditions such as asthma or chronic obstructive pulmonary disorder. These cases and many others, they need to be treated with anti-inflammatories first, such as Motrin and Tylenol. It's usually suggested to alternate the two. I'm not sure if that really is still beneficial information to do, but that's what I've always been told. And lastly, I'm going to discuss the antimicrobial peptides. They're also called AMPs. They are very short proteins that are created by a chain of anywhere between 12 to 50 amino acids. They can take over bacterial membranes by inserting themselves inside of those bacterial membranes. They then kill the microbe from the inside. They kind of work like phagocytes. These are especially important because they could be the key to unlocking the end of microbial drug resistance, which is something I discussed in one of my Build-A lectures that I did previously. Over here to the left, it's showing you how the cycle of antimicrobial peptides work. This goes around in a circle. We're going to start on one side and go around until you reach to the end there. if they could ever cure microbial drug resistance, it would be amazing. So definitely keep an eye out for these antimicrobial peptides and do your research on those and learn some more about those. Have a great day.